zusammen, ich bin Michelle, ich bin eine Biologiestudierende mit dem Mastervertiefung Systembiologie und wir gehen heute zusammen ein Systembiologie-Labor anschauen.
My name is Paola Picotti and I'm a professor of molecular systems biology at ETH Zurich. I studied medicinal chemistry at the University of Padua in northern Italy because I wanted to learn how to develop drugs. I decided to study proteins during my master thesis, and since then on I pretty much never stopped. So proteins are biomolecules that look like this. They are responsible for the majority of functions in our cells. My group at ETH Zurich studies the proteome. The proteome is the ensemble of all the proteins in our body, and we use a technology called mass spectrometry to study the proteome. With mass spectrometry you can learn which proteins you have in a given sample, for example the sample from a patient, and also how much you have of each protein in the sample. One of the recent contributions of our laboratory was the development of a method to detect altered shapes of proteins. In several human diseases, a specific protein changes shape from a healthy shape to a pathological shape, and then the pathological shape can create a lot of troubles in a cell.
So what kind of courses do you teach at ETH?
I teach bioanalytics in the bachelor, where you learn technologies to study proteins. I also teach systems biology in the masters, where you learn how to use data on the proteome to understand what is going on in a cell. If you're interested in what we do, why don't you come and have a look at our lab?
Okay, let's go.
Let's go.
So, hi, welcome. I'm Chris. I'm a PhD student in the Picotti lab, and I will show you how we look at Alzheimer and how drugs in Alzheimer patients actually work.
So in my PhD, we look at Alzheimer, and in a healthy brain, we have proteins floating around within a cell. Now, once you develop Alzheimer, the same proteins start to clump together, and they build these huge aggregates. We now add certain drugs, and see if we added some drug, do we reverse this process so that we have less symptoms.
And how do you do that?
So to do that, we use a so-called mass spectrometer, where we can measure all kinds of proteins from a brain cell.
So what exactly do you do every day when you go to the lab?
So the nice thing about my PhD is very versatile. There are some days I spend in the wet lab doing pipetting, on other days I'm at the mass spectrometer measuring samples, and also the days I usually spend on the computer doing data analysis.
Why would you recommend future students to go to ETH and study biology?
So the nice thing about biology ETH is early on, starting your bachelor, you're exposed to actual research. You're parts of different labs, you see many different labs, how they approach certain topics, how they scientifically work, and thus you have these very broad views into many different research topics.
Thank you so much for showing me around, and have a nice day.
It was a pleasure.
Bye.